away from tree building and move into the taxonomic diversity, taxonomic and morphologic diversity of mostly bacteria today. So I have this is an old tree, but what I want to point out is that when you build these trees of life and you try and represent different types of organisms, by far and large, the biggest group is bacteria. There is an enormous amount of diversity encompassed in these small little cells that all look somewhat alike. So there's a few types of diversity that we're going to be focusing on, and we'll see reoccurring themes regarding this throughout the rest of the course. So there's diversity in form, which we won't go into in great detail. A lot of the reason is because there isn't a ton of diversity in form. You may have noticed that when looking under the microscope, you see dots, especially if you've got the really small rods. There are lots of bacteria that look like that. You have lots of bacilli, cocci, bacteria that are very different phylogenetically and in terms of capabilities can look almost identical under the microscope. Functional diversity, this is really where you see tons and tons of diversity in these organisms, and we're going to spend a good chunk of the second part of the course talking about functional diversity. And then, of course, phylogenetic diversity. You can't understand functional diversity and bacterial diversity without understanding it in the context of phylogeny. So we're going to do a quick tour of diversity of form. So when you look under a microscope at bacteria and archaea, you see the types of forms that you're going to see fall into three main categories. And one of them is bacilli, or rods. And you might see a few different types of rods. You might see filamentous rods, so rods that have long filaments, and then you see more standard rods. You have the Sporilia, so that encompasses spirochetes and other organisms in spiral forms, and then cocci, circular types of cells. Sometimes you also see, especially in archaea, kind of interesting forms of cells. You can actually find square cells. You can find ones that look a little bit like eukaryotic cells. And this lack of morphologic diversity is one of the reasons why molecular work, so 16S rRNA sequencing, genome sequencing is so important in classifying and understanding the diversity that's present in these microorganisms. And of course, there are always exceptions to the rule, even though a lot of them don't look, you can't distinguish very well between many of them. There are some interesting forms that they take. And actually, by way of example, I put Anthony's picture from his isolate here. And he's not here, but this is his isolate, and you can see that it's a little bit unusual. It has these very, very long filamentous type structures, and they're branching patterns. And we actually took these pictures over to David Bermuda's, and we've been playing the is it fungi or is it bacteria game. I'm betting on bacteria. David Bermuda's isn't sure. So you do see, I think it's an actinobacterium, but the 16S rRNA sequencing will tell. A few other different examples of unusual form are shown here. So this top one, this Chrondomyces, was the bacteria that wanted to be a fungus. So it was originally discovered in sheep poop, and they really thought it was a fungus because it looks like you've got these spore packages, and it turns out that it's a bacteria. And what it does is it produces this fruiting body, and instead of individual spores being here, those are packets of many, many, many different bacterial cells, and they can be dispersed, much like spores are in a fungus, through things like wind, water, insects, that type of thing. This cell right here actually has a nucleus-like structure in it, so it has an inner membrane that surrounds the DNA. We've talked a little bit about the Thiomargarita right here, and it is large enough to be seen by the naked eye. So very, very, very few cells, bacterial cells, are large enough to be seen by the naked eye, but there are some, and here's a fly for comparison, and then a microscopic image right there. So this is another unusual example, and this is what people call and I'm saying this in quotes, multicellular bacteria. And what this is these organisms can produce fruiting bodies under extreme conditions. And it's multicellular in that it's not all the same organism, but what happens is these cells get together and there is a diversification of labor and function within the structure. So different cells are playing different roles in making this fruiting body. And this fruiting body then can disperse cells into the environment. So it's a pretty unusual type of system. You don't see this a whole lot, but it's an example of some of the unusual forms that bacteria can take. Yeah?
Do you also know the names of each one?
Nah, not for these. So another diversity of form that you see are biofilms. And I think biofilms are something that are neglected quite a bit in microbiology classes, and I'm going to be guilty of neglecting it too in this class, except for just mentioning it right here. And what biofilms are layers of different bacterial cells. So they layer on top of one another, and they are brought together or held together in place by things like exopolysaccharides that the cells secrete. And these biofilms can be super hard to penetrate by, say, like antibiotics. And these biofilms form in one of the places that they form that cause large problems are in different types of the body or different parts of the body, especially on pieces that may have been introduced from the outside. So things like contact lenses. There's a huge problem if you have, say, artificial joints or something like that of these biofilms forming. And what happens is because of this dense exopolysaccharide layer and the depth of these biofilms is the antibiotics have a really hard time penetrating the biofilms. So it can be really hard to get rid of these types of infections just because the antibiotics can't even reach the cells, even if the cells may be sensitive in and of themselves to antibiotics. So contact lenses, artificial joint replacements, dental plaque. So when you go to the dentist and they say you haven't been flossing, it's because you've got these thick biofilms that have formed on your teeth and you aren't physically disrupting them. I guess I shouldn't say you. I assume everyone in here flosses twice a day and has no issues with that. Yeah?
Is there a difference between biofilm and fruiting bodies?
A difference between biofilm, what?
In the previous slide, fruiting bodies.
Oh, yeah. So the fruiting bodies are these groups of cells that are acting in concert like an organism. So there's different cells that form different functions within it, and all of those functions are necessary for the big fruiting body to do its job. Whereas with the biofilms, they're just layers of individual cells. And they may be different species, and they may work together in concert. So like, what I mean by that is some cells, the waste products may be used as energy source from the other cells, but they're not acting together like a single organism.
Okay.
So it's quite a bit different. Yeah?
I think the fruiting bodies, from what I remember from micro, they, like if the cell is lacking in nutrients or whatever, that the fruiting body is able to provide for it, like in need of, like if it's starving.
Mm-hmm. Yeah, that can happen. Yeah, absolutely. So, yeah, and so these biofilms, what happens is they build up and they build up, and then the biofilm can be dispersed, especially in low nutrient conditions, and then the individual cells can go off and search for greener pastures, so to speak. Biofilms are also, you see them all over nature. So you go out to the environment and in lakes and ponds, you see these these biofilms. They really are everywhere. In fact, in places like soil, you're going to see a lot of, well, you won't see them, but a lot of the cells exist in biofilms. So it's, even though we often consider cells or organisms as individual cells just doing their individual thing, a whole lot of times they are in biofilms, and they can't function without the other cells around it acting together, which may be one of the reasons why, or it is one of the reasons why we can only culture a small portion of cells in lab, is because a lot of them require interaction with other species in the environment. So another diversity form is motility, and a lot of you have encountered this in your isolates. When you do a streak plate, you have a cell that lands on the agar, and if your cells aren't motile, an individual cell will divide and divide and divide, and you get this nice punctate colony. Whereas those of you who have mobile ones will try and make a streak plate, and it'll just kind of grow and spread out. So there are several different types of motility, but the one that we commonly think about are bacteria that are motile because of their flagella. And in fact, some of you in 315 will have actually learned the mechanisms of how flagella work and the way that they perform chemotaxis to get to different types of nutrients. So another type of diversity that we have is what we call reproductive diversity. And again, normally when we think of cells, bacterial cells dividing, we think of E. coli in culture, where you have two cells or one cell that divides into two cells, and you have exponential growth. But there are a fair number of bacteria that form these endospores. And these endospores are like seeds where the DNA is packed very tightly and protected, and this protection is really, really great. So these spores can survive for very long periods of time. They can survive extreme conditions. They can survive cold, they can survive heat, they can survive radiation, and they only germinate when conditions are favorable. And we'll talk about that a little bit more as we go through the course, because it turns out that they are pretty important, or very important for human health, bioterrorism, that type of. Certain species that form spores, actually this is a picture of a Clostridium difficile, can cause explosive and sometimes fatal diarrhea. So we'll talk about that a little bit. And that's why you've heard of fecal transplants. Anyone heard this? So it's this big new wonderful thing, actually, fecal transplants that have been saving lives for people with big issues with Clostridium difficile in their guts. So look forward to that explosive diarrhea lecture coming up soon. Okay, so diversity of function. This is where the bacteria and archaea, where you really start to see a whole lot of diversity. For humans, mammals, animals, there isn't a whole lot of diversity. We can't really do a whole lot. Our carbon sources are pretty boring. Hamburgers, Panda, Express. We have carbon that we eat, and we use oxygen as our terminal electron acceptor. We breathe it out. So the types of metabolisms that animals do are pretty mundane. You can't do a whole lot. But the types of metabolisms that bacteria can perform are spectacularly diverse. It's really interesting when you get down into it what they can do. So we're going to talk about the bad in terms of diversity. So many bacteria are pathogens. And I have a few examples here. And one of them up here is the outbreak of E. coli in Chipotle. Do you guys remember that? I think that was back in 2015. I'm not sure that Chipotle has recovered completely yet from that. So there were issues in the western states, and then Minnesota, and then some states, New York, Ohio. I've had issues with that, and if I recall correctly, and I'd have to ask Dr. Cooper because he's the food micro person, but it was issues with the food handling chain. So there, and then every so often you hear of these big outbreaks of foodborne pathogens that will kill people. I think one person died from this, but the big one that I remember, and one of the first that was really widespread, was back in the early 90s, and it was in Jack in the Box. Anyone remember that? Yeah, a few people have either remembered it or heard about it. So it was in the 90s in Jack in the Box, and there was an E. coli outbreak, and it killed a bunch of people. And it turned out that it was likely from E. coli contamination and them undercooking their meat. And so it was a result of these deaths from Jack in the Box that cooking hamburger became more of a focus in terms of a public health safety efforts in both restaurants and at home, and to make sure that people weren't eating, consuming live pathogenic E. coli. All right, another example. This occurred in the last year or so, and that's anthrax in reindeer. Anyone heard about that?
Anthrax virus?
Reindeer.
Oh,
So this happened in Siberia, actually not too far from where I was when I was in Siberia last summer. And people in some of these northern Siberian villages started to get sick from anthrax. And what they ended up finding, what they found out was the source were a bunch of dead reindeer that had had anthrax that had been frozen in the permafrost for a very, very, very long period of time. So these reindeer died of anthrax. Because it was so cold, their carcasses were frozen permanently in the permafrost. But what has happened is that global warming has been thawing the permafrost. You've seen all these news about the ice sheets cracking and being reduced. Permafrost is also thawing, and these reindeer carcasses thawed. And the thawing of these reindeer carcasses that had died due to anthrax, now the indigenous people were being exposed to them, and they were dying. And anthrax produces spores, anthrax produces spores, and they're very robust spores, and so the people came in contact with these newly thawed reindeer carcasses, and they were infected with anthrax. So this right here, anyone know what I'm referring to right here? When I have these three pictures together? Anyone take a guess? What really fun disease? No one? Plague. So the Black Death. And some of you have probably seen pictures like this, right? Maybe? So you're nodding. So during the plague, what would happen is that there were certain individuals who were designated as plague doctors. And so what they would do is they would dress in these large cloaks, not costumes, but large cloaks and protective clothing that would theoretically prevent exposure to whatever was causing the plague. You know, we know now, of course, it's bacteria, but they had no idea then. And then the reason for this beak-like mask was because they, in front of their mouths, wanted to pack in things like hay and herbs and smelling salts and those types of things, which they thought would help protect them from disease, but then also filter out the really terrible smell of death and decay. So when you see pictures like this, they're really super creepy, but it's a picture of the plague doctors. And you know, they'd call in these doctors, and really they couldn't do anything. They could not do anything to treat people with plague. What they often ended up doing is just keeping accounting of just how many deaths had taken place. So eventually it was figured out that plague is caused by a bacteria, Yersinia pestis, and the bacteria is indigenous. It's found in rodents. And the way it's transmitted is not from person to person. So chances are, if one of these doctors did come into contact with someone with plague, they weren't going to get plague from that person. The way it was transmitted is through fleas. So you'd get into an environment like medieval times, and you'd have rats, you'd have rodents, they'd have fleas, and the plague would be transmitted by flea bites. So one thing that people don't realize is that plague is present in the US. I wouldn't be surprised, no one's tested this, don't freak out, I wouldn't be surprised if some of the squirrels on campus had it. There were a few deaths in Yosemite several years ago from the plague. So what happened is that rodents in Yosemite carry it, and people perhaps went and messed with the dead bodies of rodents, or they camped too close to rodent burrows. And so what you see here, and this isn't showing up very well on the projector, is they closed several of the campgrounds, including Crane Flats and my favorite campground, Tuolumne Meadows. And they went in and they treated the rodent burrows for fleas. And now if you go up to Yosemite, all over the bathrooms, you'll see signs that say, be careful, there's plague here, don't mess with rodents, which is a smart thing anyway. So then finally, the other example that I'm giving here is raw milk. Raw milk is milk that hasn't been pasteurized. So pasteurized milk is essentially milk that's been heated up to high temperatures to kill any bacteria that lives in it. So there's this movement of people who prefer to drink raw milk, so milk that hasn't been pasteurized. And the problem is that there are bacteria in even if you drink raw milk from a safe source, there will be instances occasionally where there's bacteria like listeria in it and you can be infected. So my granddad grew up on a cattle ranch, and he always drank raw milk as a kid. And my dad spent summers, and he became an accountant eventually, but that's another story. And my dad would spend summers there, and he drank raw milk and really liked it. So I'm not going to advise you on whether or not you should be drinking raw milk, but I will mention that sometimes, on occasion, my family will drink it as a little bit of a treat, but even my granddad and my dad, who have had lots of experience with raw milk, have decided not to drink it on a regular basis. And you certainly shouldn't drink it if you're pregnant, immunocompromised, and you shouldn't let little kids drink it. So that's what I'll say about the raw milk debate. The last outbreak was from a organic farm, so a farm that had good practices, and it's just something that happens. Yeah.
What about almond milk?
Almond milk's fine
Okay, 'cause why?
if it's been pasteurized. Pasteurization is the key. Though with almond milk, I have to say, I don't think almond milk has to be pasteurized, but I'm not sure. I'm not sure. I guess it depends on the process by which they take the almonds and process them and get them into milk form. So they may pasteurize it to increase the shelf life, but unlike the dairy industry where you have exposure, you've got dairy cattle, exposure to all sorts of bacteria on a dairy farm. Of course, almonds don't have that. So I guess I'm not familiar enough with the regulations to know.
Dr. Sue, he spent like 30 minutes on his class talking about not to drink milk.
Not to drink milk at all? At all?
Professor Basu told us the same thing.
Told
Told
not.
us not to drink milk from other animals.
Oh, from other animals?
Yeah. He said the only milk that we should be getting is from the mom, from our moms.
Oh.
Whenever we're a child.
Yeah.
That certainly. So he's advocating not to drink milk at all as an adult. That's one opinion. So
Well, he was just pointing at the fact that many people are lactose intolerant, and he said we're just not meant to. We don't have the genes for drinking.
well, that is where I can correct him a little bit. So this is another completely interesting story, but since you brought it up, I want to make sure that the pertinent information is there. Okay, so the ancestral state for humans, so ancestral means, you guys now know what the ancestral state is. The ancestral state is for lactose intolerance. That is that we do not break down the milk sugars after we've been weaned, okay? And it's not because we don't have the enzyme. What happens in the ancestral state is that the gene that produces the enzyme that lets us break down milk sugars is only turned on in babies, okay? And so ancestrally what happens when humans get older is that gene's turned off. So in adults, even though the gene to produce that enzyme is there, it's no longer expressed. So everyone on board with that? That's the ancestral state. What happened in some ethnicities, not all, some Northern Europeans, the Maasai in Africa, this happened with. So groups of people who were exposed to dairy products, so raised cows, like the Maasai famously have their cattle that they raise. Northern Europeans had lots of dairy. What happened is that there was a mutation, not in the gene, but in the promoter of that gene that allows us, I shouldn't say us, I'm not lactose intolerant, so me and other people who can drink milk without problems. Essentially what it has made is so that people can produce that enzyme through adulthood. And it's a mutation, not in the gene, in the promoter. So there are a lot of us now who produce that enzyme and have no problem with dairy. And so it was an evolutionary adaptation to allow us to be able to utilize essentially the calories and the nutrition that were available. In the case of Northern Europeans, certain African cultures, I'm not sure where else this occurred. We are genetically now able to do this. I know a lot of Asian people aren't able to. I think there are parts of India where they can't. I'd have to look at the population demographics to see exactly where this is coming from. So some of us are genetically made to do that, and it's a recent adaptation. And and if you're lactose intolerant, it's because you can't produce that enzyme into adulthood. That make sense? Kind of an interesting story. What's kind of cool is, okay, this is an example of convergent evolution too, because it occurred independently in a bunch of different human populations. So the mutation that, say, allows Northern Europeans to produce this enzyme into adulthood is actually different from the mutation that allows the MASI to do it. But it's in the promoter, and it does the same thing. It's effectively allowing this gene to be turned on into adulthood. Yeah?
There was a chapter that talked about that in the book that Shabbat made us
Uh-huh.
do a report on, and it said that people who aren't lactose, who have the gene to digest lactose, that they have a reproductive advantage because they can get more nutrients from different sources.
Uh-huh.
So that's why it's like stay remain, and even like people with admixture, like everything else,
Uh-huh.
like they will most likely get like it'll be like a dominant.
Yeah.
Yeah.
Yeah, exactly. So yeah, I mean, that's exactly why. Because, you know, if you think back to early, 10, 15,000 years ago, 5,000 years ago, food was not so abundant. And so if you could get extra calories, that was a huge advantage. And, the reason it's dominant, so like, let's say you have mixed heritage, and Northern European, and then from another ethnic group that is generally lactose intolerant, you'll still have the one allele that allows you to produce that enzyme. So you may be slightly less, or you may be a little not as tolerant as some other people. Like you may offend your spouse if you eat a little bit too much, drink a little bit too much milk. But that's why it would behave as a dominant trait. Yeah, Anthony?
So you said like two ethnicity, like you said it was convergent. You mean like they both have the same or same mutation that allow them to or the same mechanism?
Yeah. Well, so let me make sure I understand your question. So like two different groups that can do it.
Yeah, I mean what you say is like so one of them is the promoter that's been mutated.
Yeah.
And the other is that the same thing.
Both of those groups, and the examples that I know specifically about are people of Northern European heritage and the Maasai. Mutations occur in the same promoter in both groups. It's just different bases in that promoter. So, say in Northern Europeans, you have a mutation that's in a specific base. It allows the gene to be turned on. In the Maasai, there's a mutation in that promoter. It's a different base, but it still allows it to be turned on. So that would be an example of convergent evolution. So anyway, that was a bit of a detour, but I used to do human genetics, and so it's, I still like to keep my fingers in it a little bit. And that's a particularly interesting example of recent human evolution, so to speak. Yeah?
Oh, I was going to ask this. You said you used to do human genetics.
Yeah.
So I have a friend who was lactose intolerant.
Uh-huh.
She started like self-dosing with other doses.
Uh-huh.
She's brought herself up to the point where she can actually drink them.
Uh-huh.
So I was wondering, is there like something else that regulates this gene being turned on?
That's entirely possible, but I don't know enough about it to comment on mechanism. And with anything, you can build a tolerance over time. Like, I can drink more caffeine than you would expect Dr. Cooper to be able to drink and function. Yeah, and still go to sleep at night, but if I drink alcohol, I'm like, after a beer, I'm asleep. So, you can build a tolerance. So. Okay, other questions after our little detour? But that was a great detour because we were able to bring in some phylogeny, convergent evolution, that type of thing. And it's super interesting. Okay, we'll also talk about the good. So bacteria, and this has come to the forefront more and more and more lately. You've probably seen more stories about the microbiome in the news. And one of the most important things to come out of the microbiome studies is it's enormous important in our guts, in our health. So as we go on through the semester, we'll talk a little bit about the role of the microbiome in our gut and the different ways that it influences human health, because it can, in very significant ways, impact human health. I tossed in an example of the rhizosphere in there because, as you know, the bacterial symbionts that inhabit the roots are very important to a plant's health and therefore important to feeding the human race. Okay, and the unusual. And I made a mistake in here. This should be high pH, but these are some examples of really extreme environments where bacteria and archaea thrive. So this is an example of a boiling acid pool, essentially, that bacteria and archaea can survive in. You have these salt ponds where water evaporates and you have salt, very, very high concentrations of salt. You can have mostly archaea that survive in there and produce these interesting pigments. Like if you fly over the San Francisco Bay, you'll see that. And the pigment is due to different microbes in there. Again, this should be high pH. This is very basic. Anyone know where this is? Anyone know where this is? It's Lake Mono, which is up by Yosemite. And so this is an alkaline, very alkaline lake, and you go there and it's super stinky. And they found really interesting organisms there. And then I had to throw in some below freezing environment because they're microbial communities that survive below 0 degrees Celsius. So all sorts of these crazy unusual conditions can be inhabited and by growing, thriving bacteria and archaeal communities. Yeah?
High pH? Because like otherwise.
Yeah, that's what I said. It should be high pH.
Oh, okay.
Yeah. Yeah. So that was my mistake. It should be high pH because it's basic. Yeah, let me do this. That should be high pH. And another thing that people neglect in microbial not microbial ecology, but microbiology type classes is the fact that microbes run global biogeochemical cycles. So the carbon cycle, the nitrogen cycle, these are run by microbes. And people are realizing that more and more, which is why microbial ecology is such an emergent field right now. And we'll talk about this quite a bit as the semester progresses. Okay, so let's talk about phylogenetic diversity. And we'll link phylogenetic diversity to some functional diversity as well. And so my big question was, how was this done? How did they start looking at phylogenetic diversity?
16S.
Yeah, 16S rRNA sequencing, other ribosomal subunit rRNA sequencing. So very much a genetic molecular-based approach. And this tree right here is a pretty decent tree of the major bacterial phyla. And we're going to, as we move through this lecture and the next lecture, we're going to keep coming back to this tree so that when we talk about different groups or different phyla of organisms, that we can place them on the tree and understand them in the context of how they're related to everything else. This is a different view of the tree. And actually before I move on, what I want to say is about 12 years ago, so not that long ago, about 40 different phyla of bacteria were recognized. Now there are 200 to 250, even 300 phyla, and many of these phyla are only known because of DNA sequencing. We haven't actually cultured them. Which brings me to this tree. This tree is obviously an unrooted tree, and it represents some of the major phyla of bacteria. The things that you'll want to note in this tree are the different colors. So in blue, and let me zoom in a little bit to some that you know and love. Okay, in blue are the phyla where there are cultivated members. So members that have been cultured, studied in lab. Here's the proteobacteria. Here are firmicutes, cyanobacteria. So some of these phyla that you have seen the names of and you might be familiar with. So that's everything in blue. In red, and you can see that red represents a substantial portion of the tree. And in fact, there's this whole group over here with no cultivated members. So red are phyla with no cultivated representatives. We only know about them because of genetics. And then there's this green dot. And what the green dot is, the green dot, let me find one. Let's look at this group here with no cultivated organisms. Despite the fact that a lot of these haven't been cultured, some of them have complete genome sequences. Not 16S rRNA, but complete genome sequences. And what people have started to do, this is really new, it's really cool. What people have started to do is they've started to get really sophisticated cell sorters. And what it can do is you can take a plate, let's see, I don't have an example here, but, well, it's the equivalent, let's say you can take some tubes and you can sort the cells such that one cell lands in the tube, just one cell. And people have gotten to the point where they can actually amplify the DNA from that cell and sequence at least part of the genome from the DNA from one single cell. This is like super sophisticated, super cool technology. So what you're seeing in the green dots are the organisms where the only reference genome is from a single cell, from that particular phylum. So we have whole phyla of organisms where we only know because we've sequenced the genomes of a single cell. So it's, I think that that's really pretty interesting, and it tells us a whole lot about the enormous diversity of life that we know nothing about. Yeah? Well, okay. So let me step back a little bit. So for all the blue, there are multiple organisms that have had their genome sequenced. Okay, so we have the genome sequence of E. coli. We have the genome sequence of different Shigella. We have the genome sequence of all sorts of bacteria. It's actually easy to sequence bacteria genomes. So the problem, and what you do is you grow them up in culture. So you have tons and tons and tons of cells. And you extract the DNA, and then you sequence the entire genome. Not just the 16S, but the entire thing. Not a problem. Not a problem with technologies these days. That technique requires that you be able to grow these cells in culture. So a lot of these, like especially this group right here, you can't grow in culture. So you can't do that. You can't grow up a whole bunch of cells, extract the DNA, and sequence the genome. So what people have had to do is they've had to take a method where they can use a machine to separate the cells out, let's say you went into the Gulf of Mexico and got a sample, and you wanted to sequence organisms in your sample, but you couldn't because you couldn't grow them. So what you would do is you'd put it through this machine, this cell sorter, that would make it so that it could sort cells, so it could put one cell in a tube. So you have this tube with one cell, and people have developed methods that you can sequence the genome from that one single cell, whereas traditional methods require that you grow up cultures with enormous amounts of bacteria in it. So it's really, really cool that you can sequence the genome from a single cell. It's like science fiction. So everything with the green dot are phyla, where the only reason we have a genome is because we've sequenced it from a single cell. We can't culture anything from those phyla. Okay, that make sense now? All right, any other questions on that? So the point of this is that the tree of life, super diverse, we know nothing. or we know very little about it. There's still vast amounts of information that we do not have. As a reminder, when we're talking about phylogenetic diversity, we have our domain, which is bacteria. So we talk about the 3 domains of life, right? Bacteria, archaea, eukaryotes. And then we go to the phylum level. And note that there's no kingdom. They're not teaching, I don't think, kingdom anymore. I hope not. When I was an undergrad, they were. But we don't really talk about kingdoms anymore. So I'm sure there's a mnemonic to help you remember it. I don't remember the mnemonic. But domain, phylum, class, order, family, genus, and then species. So you can see the organization here. And I have this set up so that it goes through and you can recognize some of your favorite organisms. So proteobacteria is a really big phylum. And you can go to proteobacteria and go to the class, let's say gamma proteobacteria. And if we go to the order in Enterobacterales and then Enterobacteraceae, which is the family, you get down to the genus and you see a lot of things that should be pretty familiar. So this one genus in the proteobacteria group has Escherichia, so E. coli, Klebsiella, you should probably recognize Klebsiella from 315, Yersinia, so Yersinia pestis, causes plague, which, by the way, one reason why I'm not very afraid of plague is because it's so treatable by antibiotics. I mean, this entire epidemic from medieval times that was so deadly, easily treatable nowadays. Here's dysentery, all sorts of nice things just in this one group of organisms. And let's stop right there. What we're going to do in the next class or two is we're going to talk about some of the major phyla of bacteria that you hear about over and over and over again. The proteobacteria, the firmicutes, the spirochetes, chlamydia, those types of things that you should be familiar with after taking a diversity course. So, questions? All right, let's meet back at 2 for lab.